Our second speaker today is Dr. Sandra Irmisch. Dr. Irmisch is currently a research associate at the University of British Columbia in the lab of Jörg Böhlmann. Sandra came to UBC as a Theodor Linen Fellow of the Alexander von Humboldt Foundation. And according to the information that I got, she's managed to secure a PI position at the University of Leiden starting in 2021. So congrats on that. Sandra's work has been looking at the natural products that are produced in plants, in particular things in the flavanol glycoside pathway. And she's done some really heroic work of reconstructing biosynthetic pathways for something called montbretin A. And that is, I believe, what Sandra is going to talk to us about today. So without further ado, Dr. Irmisch, the floor is yours.
Hello, everybody. And I want to thank, of course, the organizers of the GlycoNet seminar series for inviting me to give a talk here. I want to start out with a little disclaimer. I'm not a biochemist. I'm a molecular plant biologist by training, as you just heard. And therefore, the talk I'm giving today might be a little bit different from what you might have expected, but I promise I take you on a really exciting journey from a plant to the biosynthesis of a specialized metabolite, a glycosylated metabolite, which has great application in the treatment of type 2 diabetes. Okay, so a lot of you guys in the audience are probably chemists, and you're probably really good in the lab synthesizing small molecules. And I don't want to step on anybody's toes when I say, you can try as hard as you want, you're never going to be as good as my study organisms. And my study organisms are plants. So plants, in my humble opinion, are the masters of biochemistry. They synthesize a vast variety of different small molecular compounds, and some of them you see here on my slide. So they're small molecules, they differ in complexity, in stereochemistry, and plants have the ability to produce them really precisely. There are over 200,000 different structures known, and the reality is probably more like there are more than millions out there. We just don't know yet. Plant biologists call them specialized metabolites or secondary metabolites. And this comes from the fact that they are not absolutely necessary for the plant's survival, but they certainly give the plant an advantage in its day-to-day life. And I don't want to go into detail, but I want to mention it because it just like is for the complete picture here that plants do produce those compounds in order to deal with their daily life. So it is for their protection against UV light, for example, or to fight off herbivores which might eat the plant, or to even communicate in their community. Okay, and we as humans, we have been really smart because we actually utilize plants because of those small molecules since centuries. And we do this because those molecules do have different flavors, they have different fragrances, so we use them as in our food or in our perfume, and they also have different functions. And some really, really well-known examples are those two molecules. So I'm showing you caffeine here and nicotine here. And those are molecules you consume quite often. So coffee, maybe even now, maybe you're sitting in front of your computer trying to stay awake during my talk and consuming caffeine because of its stimulating function. And the same goes for people who like to smoke a cigar for the nicotine, for example. But it's not just that we enjoy to consume those molecules. The vast area of plant specialized metabolites also give us some of our most important medical compounds. And I just want to mention a few here. So we have morphine, which is originally derived from the latex of the opium poppy, for example. And morphine, as you might know, is an anti-pain medication. And then I want to mention Taxol. Taxol, a really important anti-cancer medication, which is originally derived from the bark of the yew tree. And if you look at both of those molecules, they are really complex and they're hard to synthesize by normal chemical means. And then I want to introduce you to another compound which has quite medical application because it has been found to be an anti-diabetes drug. And this compound is called montbretin A. So here you see montbretin A. It is a plant-derived compound and is quite complex. It's an acylated flavanol glycoside. So I'm going to explain the structure to you. So we have a flavonol core here, which is a myostatin. And then we have two sugar chains attached to my myostatin core. So we have a trisaccharide on the free hydroxy of that myostatin here, consisting of a rhamnose and two glucose units. They are 1,2 linked. And then the middle glucose on the 6th position has a caffeose chain attached to it. And then we have a second disaccharide chain on the 4' position of that flavonol 4' hydroxy group. And this consists of a xylose and a rhamnose linked in one form. Okay, this is a really complex molecule, and we can make it a little bit easier for the eye if we just depict it as the building blocks it's made of. So this is going to be what I will use for most of my talk, just like the building blocks, the myostatin, the different sugars, and the caffeine moiety. Okay, as I told you, montbretin A is a potential new drug for the treatment of type 2 diabetes. And you probably all are familiar with diabetes. It's a worldwide epidemic. It affects over 400 million people, and the number is rising because it is due to our lifestyle. It is due to us eating too much sugar, being too lazy, not moving as much as we used to. And so we really need new medications to treat diabetes. Type 2 diabetes is due to the body's inefficient use of insulin, and this leads to elevated blood glucose levels. And if you have elevated blood glucose levels over a longer period of time, this has detrimental side effects such as kidney failure, heart failure, blindness, and automatically leads to premature death. Okay, so how did we come to actually find that compound? So montbretin A was actually discovered here at UBC. It was a research project which was shared by 3 PIs here and under the lead of Stephen Witters, which you might know. And they discovered montbretin A when they were looking for a new type 2 diabetes drug and actually screened a big natural product library out of 30,000 different natural product extracts containing many extracts of plants. And montbretin A works in a way that it actually reduces postprandial blood glucose levels by inhibiting the human pancreatic alpha amylase. So what does that mean? So reducing postprandial blood glucose levels means it reduces your blood glucose after you've eaten something. And in our Western society, our like a lot of the glucose we're actually absorbing comes initially from starch. So our diet is just like really rich in starch. So what happens when we eat starch? It gets digested in our intestines and digested over several steps into monomeric glucose units. And those glucose units then get absorbed in our bloodstream, leading to elevation in blood glucose levels. So if you're healthy, this doesn't really affect you. However, if you suffer from type 2 diabetes, this initial blood glucose spike you're getting after eating something actually affects you because your body cannot regulate it down. So what montbretin A does now is it actually inhibits the initial degradation of starch. It inhibits the human pancreatic alpha amylase, which is the first enzyme in starch degradation. It does so really specifically and really effectively. So we have a Ki here of just 8 nanomolar. And by inhibiting starch degradation, we actually get less glucose, and hence our blood glucose levels stay low. So this is the way MBA actually functions in helping people who suffer from type 2 diabetes. I told you in the beginning that MBA is actually a plant-derived metabolite. So let's have a look at the plant it actually comes from. And this is where my area of research starts. So it has originally been found in a plant which is called Montbretia or Crocosmia crocosmiflora. This is this nice flowering iris plant you see here. It's originally from South Africa, and montbretin A has been detected in those underground storage organs, which are called corms. So you see a corm here. So they're like bulb-like organs, but more distinct than bulbs. Okay, so if we now want to establish montbretin A as a treatment for millions and millions of people worldwide, we actually need to make sure that we do have a lot of the compound available for drug development and for application. If, as a chemist, you look at the structure of the molecule, you will realize it's not really feasible to synthesize that molecule by chemical means. It's far too complex, has far too many sugars, far too many hydroxy groups. So chemical synthesis is not really an option. We can isolate that compound from the plant which produces it. However, you have to take into account that the plant montbretin has never really been cultivated for cultivation purposes. So it has never really been optimized for growth, for MBA content, and so on. Also, the plant doesn't really contain that much of the compound, and we're talking about the below ground reproductive and storage organs of the plant we would be harvesting. So it's kind of a distracted harvest. So at the moment, and also inside of like having a sustainable production system and plants which are always affected by climate change, using a plant system to really produce montbretin A is not really an option either. And this is where our research starts. This is where we want to actually engineer an alternative production system for montbretin A to really make sure we can produce it in high amounts and in a sustainable way. But how do we actually do this? How do we get from a plant to the production of a molecule? So our aim is to actually take what the plant does and put it into something else so that we can just mimic biosynthesis somewhere else. And the way we are trying to do this is with those steps, and I'm going to go through them in the remaining of my talk step by step. But just to give you an outline, so we usually start with a good metabolomic profiling to get an idea what's actually going on in the plant. Then we need to get some sort of sequence information, genomic information, transcriptomic information, or even proteomic information, which then actually guides our gene discovery and ultimately our enzyme and pathway analysis. And this will then feed into the synthetic biology approach and the metabolomic engineering ultimately to reconstruct MBA biosynthesis somewhere else in order to facilitate drug production. Okay, but first thing first, let's start right at the beginning. So those are montbretin plants growing in the field. They look really lovely. Apart from just using them as cutting flowers, however, no science has been done on that plant ever before. So we really had to start from the basics because there was literally nothing known. But where do you start? You start with answering basic questions about where and when is montbretin A actually synthesized in montbretin. And for that, we of course looked at the whole plant, and we found that the corms are actually the organs which contain the most montbretin A. And the corms are the overwintering reproductive and storage organs of the plant. And because they are the reproductive organs, we decided to have a closer look at those corms over the period of a whole year. And this is what we saw. So we planted a corm. In Vancouver, we usually do that around February. In Alberta, you probably do that later on. And when you grow a corm, it grows stolon, and then it will form vegetative tissue above ground, ultimately flowering. And on the bottom here of that vegetative tissue, this is where the magic happens. This is where a young corm forms each year. So just to recap, you always have that old corm, which you originally plant, and then there will be, within the summer, during the growth, there will be young corms formed, let's say 5 per plant or so. And of course, we took all those corms. This is how they look like. So they start out, the young ones, really tiny, and then they grow in size and diameter. The old corms look sort of the same all the time. And we, of course, analyze them for their metabolites, especially inside of how their MBA content is. And this is what we saw. So we saw that the old corms, they don't really vary in MBA content over the year. However, the young ones, they actually have way more MBA per milligram per gram fresh weight in the summer months. And then they show this drop. And this led us to believe that MBA biosynthesis is actually active in those young developing corms, stops probably within August. The corm still grows, hence we see that dilution effect. But really for you guys, the take-home message from this slide is that montbretin A biosynthesis is active in young corms, but probably not in old ones. What we also looked at when we analyzed those corms was whether we could spot some intermediates accumulating from MBA biosynthesis. So when we look at the molecules, so we have all those building blocks like the sugars, the caffeine, the myostatin, and we don't really know how we get from those building blocks into the assembly of that complex MBA molecule. So we looked for intermediates, and we also did some crude protein assays, and we were actually fortunate enough to be able to reconstruct MBA biosynthesis. So this was what we postulated, just based on intermediates. We found what is going to happen. So we start with myostatin, and then we attach a rhamnose, a glucose to form that disaccharide. The acylation is actually the third step which is happening, and then we add the terminal glucose, and then the disaccharide on the 4' hydroxy group is added the last. So we have a 6-step linear pathway we predicting happens from the building blocks, myostatin, the UDP sugars, and the caffeine-CoA. Okay, so we did our homework. We knew MBA biosynthesis is happening in young worms. We had an idea how it's actually happening, how we actually get the assembly of the building blocks done. So now we were actually in the position to ask the question, so what are the enzymes, what are the genes now which actually drive montbretin A biosynthesis? So in order to do this, so I said we did our metabolite analysis, and what followed was that we actually create sequence information. So montbretin is not a model plant, so we don't have a genome available, and there was no other sequence information at that time point when we started that project. So we did Illumina sequencing and created our own reference transcriptome. And here I want to point out that the transcriptome always just contains the genes which are expressed in the very well moment. So you really have to make sure you're sequencing something which is also actively expressing your genes of interest. Hence, we sequenced those young worms where we before made sure they are actually actively synthesizing montbretin A. If we would have sequenced the old worms, we would have not been successful in finding the genes responsible for montbretin A biosynthesis. Okay, now we had our reference transcriptome, and then we could go on and actually select candidate genes. I'm showing you here the molecule again. And what you have to know for plants is that plants, they frequently produce flavonoids and UDP sugars and caffeine-CoA. So this is something which is like ubiquitously found in plants. However, the genes which are responsible for actually assembling the montbretin A molecules, those are the ones we were particularly interested in because those are the ones unique for the montbretin plants. And if you look into the literature and look for genes which are, or enzymes which are involved in glycosylation and acylation in plants, you will find out that we're most likely looking for UDP-dependent glycosyl transferases, or short UGTs, or bad acyl transferases, or ATs. And this is what we did. So we looked in our reference transcriptome for UGTs and ATs, and we actually found a lot. So we found about 160 UGTs and about 60 ATs, and this is too much to work with, right? So you cannot possibly just go on and like clone and characterize about 200 genes. I'm showing you the phylogeny of the UGTs here. So we did some work and could use up to a certain point the phylogeny of those UGTs and in relation to what other UGTs have been characterized from plants. However, I want to say that this is really difficult because plants do a thing which is just called gene duplication a lot in order to involve new metabolic classes. So you often end up with genes which are highly similar but do catalyze totally different reactions. So actually just going by gene sequence, protein sequence, and even phylogeny only brings you little success. We don't have a genome, and plants also usually don't cluster genes. So this is another thing. So there's nothing we could do in that direction. However, what works really, really nicely for plants is that you actually look for co-expressed genes. So the idea here is that genes which are actually involved in the same pathway, they show the same expression pattern. And here again, we used our information from the time course experiment I showed you in the beginning, the one where the young worms had really high amounts of montbretin A, and then towards the autumn it dropped. And this is sort of the expression pattern we would expect for genes in montbretin A biosynthesis. And this is what I'm showing you here. So this is a gene expression map, what we call heat map. Red color means the genes are highly expressed. Blue color means they are not as high expressed. And then down here you see June to October. So this is the growing season. And I boxed the genes which show a pattern we would be interested in. So really high expression in those early summer months and then dropping towards the autumn. And those would be genes we would use as our candidate genes to then carry on and characterize further. In contrast, for example, to say those genes over here, which show the reverse pattern we would not be interested in. And that way we could actually narrow down our target genes to like a reasonable number to actually work with. And then we went ahead and actually cloned out the genes, heterologously expressed them and characterized them. For characterization and heterologous expression, we're usually using E. coli, sometimes Saccharomyces cerevisiae and Nicotiana benthamiana, and I will talk a little bit about that later. And then product detection we usually do with liquid chromatography, mass spectrometry, and also UV. And I show you as an example, one of our reactions we detected. So we knew the first reaction in MBA biosynthesis is the attachment of a UDP rhamnose to the myristoyl core to form myristoyl-free rhamnocyte. And UGT77B2 actually had the required activity. So here you see we expressed that protein in E. coli and purified it. I'm showing you the UV chromatogram here. You see the myristoyl, and only when we have UGT77B2 present, the myristoyl is converted into myristoyl-free rhamnocyte. And using this pipeline, we were successful in actually characterizing the complete pathway of MBA biosynthesis. As we expected, it's a linear pathway. It's a stepwise assembly of the building blocks into the complex molecule. And we need 5 UDP-dependent glycosyl transferases for that, and 1 out of 2 acyl transferases. And with those enzymes in hand, we actually made a huge step towards now going ahead and thinking about actually engineering an alternative production system for montbretin A production. So just to sum that small part up before I go into the engineering, so what we did is pretty much, we did ask the plant, how are you producing montbretin A? We took the genes the plant actually uses to produce montbretin A and characterized them. And now we have the toolbox available in order to take the toolbox somewhere else. This could be a microorganism, or it could just be another plant, in order to apply synthetic biology and metabolic engineering to come to our aim, which is a sustainable large amount of drug production. So we're doing a lot of work at the moment on engineering MBA into a heterologous system. We do work with microbes as well, especially yeast, for a variety of different reasons. I'm not going to talk about this today. I just want to say that engineering something in yeast, in contrast to engineering something in a plant, has one disadvantage, and this is that you need to engineer much more. So yeast does, for example, not produce flavonoids. It doesn't produce a lot of the UDP sugar. So you have to invest much more engineering in order to convince yeast to produce the molecule for you. While working with a plant system actually has the advantage that, as I said, a lot of the molecules we need as the building blocks, they are ubiquitously found in plants. So our idea was to use another plant with the aim of actually just hijacking those building blocks it already produces and just channeling into montbretin A biosynthesis. So we're working with Nicotiana benthamiana, which is pretty much tobacco. And as I said, we are hoping to use those endogenous building blocks in order to recreate montbretin A biosynthesis by just using our MBA assembly genes with that. And the other really big advantage about working with Nicotiana benthamiana is that there is something called a transient expression system. And what you just do is pretty much you put all your genes of interest into Agrobacterium, and then you infiltrate it in Nicotiana benthamiana leaves. You wait 3 to 5 days, and then the plant actually produces your genes, your proteins, and hopefully your metabolic product as well. So this is what we did in the beginning. We just thought, okay, let's throw all those 6 MBA biosynthetic pathway genes into Nicotiana and see what comes out. And this is what comes out. It's not montbretin A, but it's really close. So we have the same sugar configurations, the same sugar. The sugar attachment works really well. The only difference here are 2 things. We have a camphoral core instead of a myristoyl core. So we're missing the 2 hydroxy groups on the flavanol B ring. We don't know whether that actually matters in inhibiting the human pancreatic alpha amylase. There is some crystal structure data which suggests it might, but there's no data for actual proof, so we have to wait here. And then instead of a caffeine group, we have a coumaryl group here. And this is the bigger problem, actually. So the coumaryl is missing that meta hydroxy group on the phenol ring here. And this hydroxy group is actually really, really essential in inhibiting the human pancreatic alpha amylase. It actually directly interacts with the active site there. So we really need that hydroxy group. So furthermore, we were thinking, okay, maybe we can convince the plant to just give us different precursors. And we started doing this by the hopes in changing that camphorol core here into a myristoyl core. And we did this by, again, going to the plant and asking the montbretin plant, how do you actually produce so specifically that myristoyl core? And we found that if we infiltrate 3 of the montbretin genes into Nicotiana, we can actually convince Nicotiana to produce that myristoyl core instead of having a camphorol core. This is a transcription factor, and then 2 genes which are involved in producing the flavanol. And it not only did change it from camphorol to myristoyl, it also increased the production of the metabolite by over 10,000-fold, which is great. And then if we now add our montbretin A assembly genes to those other 3 genes and co-express them in Nicotiana bentamiana, we can actually for the first time get the expression of montbretin A in tobacco plants. So we have the right structure here. I have to say those are still small amounts. We still have to tackle the question of that hydroxy group here. So even though we're seeing MBA, we still see a lot more MBB. This is what we call the metabolite when it has a coumaryl instead of a caffeine group here. But we're pretty well convinced that we can just apply the same principle as we did for engineering the myristoyl core and also engineering the caffeine side chain in order to be able to produce with little effort the molecule in larger amounts. Okay, with this, at my end, I hope I could give you a little bit of an insight on what we are doing with taking something valuable the plant produces in order to use it for ourselves and engineer a system which hopefully brings us in the future a sustainable production system to produce a highly valuable plant-derived compound in the fight of type 2 diabetes. With this, I want to thank all the people who have been involved with the project over the years. This is the complete Bowman lab, especially Jörg, my boss, for letting me work on the project, and then like students, technicians, and so on who have been involved. Big thanks to all the collaborators. I specifically want to mention the lab of Stephen Witters here, who has been extremely helpful and has been a really good collaboration. I want to thank the funding agencies, especially GlycoNet, as I said in the beginning, you for your attention. And let me know if you have questions.